Yeah, so I'm heading the BALDER project, which is about Bayesian analysis of diabetes for enhanced biomarker and drug target investigation or identification. And the aim of the project is to improve statistical power to identify genetic variants associated with type 2 diabetes, which can help us to better understand the underlying disease biology and subsequently also be used for identification of potential drug targets. And what is unique to this project is we will be doing this by developing and applying some multi-trait and multi-component Bayesian linear regression models that combines information from correlated traits and diseases on one hand, but also use functional information about, so basically group of genetic variants located within functional units such as biologic pathways. We have a great project team. We initially, we had difficulties in recruiting people, but now we have a very unique group of people that have different skill sets that complement each other very much. So we have Marina, Victor, Tahiria, and Xiong Hao from the Center for Quantitative Genetics and Genomics. We have Astrid and Mads from Department of Biomedicine, also at Aarhus University. We have Palle from Department of Health Science and Technology from Aalborg University. And then we have Joanna Hausen and Sil Hu from Novo Nordisk Research Center, Oxford. And always in the background, we have Marie-Louise and Ditte Høyer, who is very much a support for us, both from the beginning, but also during the project. So we appreciate that very much. So it's a focus here is on the disease type 2 diabetes, and we have a background in genetics, so we are very much interested in the genetics of type 2 diabetes. And one of the characteristics is that it has a high heritability. That means, and that's important both for understanding using genetics for understanding the biology, but also for developing risk prediction models. What we also know about type 2 diabetes at the moment, it is influenced by a lot of variants that each only explain a very small proportion of the overall heritability. And another characteristic is that the variants are not randomly distributed over the genome, but they do tend to be enriched in biological pathways and other functional units on the genome. Another important aspect of type 2 diabetes is that it's highly correlated or strongly correlated with a range of other traits and diseases. And all of this information we have about type 2 diabetes, we can use to actually in the model development. So basically, what we, of course, we could always collect more data, but what we are doing here in this project is actually we develop some methods that allow us to use the data better. And that is both using correlated trait information, but also using the prior information in the functional marker groups. The BLR models, it's a methods model that's proposed for as a very unified approach to map genetic variants, to estimate genetic parameters and prediction the disease risk. It handles the different genetic architectures in a very flexible way. It scales to large data, and we have extended to multiple traits and multiple marker sets. And it's implemented in our new release of our open source software package called QDG that's available on GitHub and Crane. Here's just a figure illustrating the first step in the modeling approach. So basically to the left here, we have standard geo summer statistics from, so that's basically we have a lot of association signals from for many genetic markers, millions, in the millions, we can also have information for many correlated traits. What we then do, we put it into this BLR model machinery that take advantage of two types of correlation. One is the genetic correlation between traits, and the other correlation is the the correlation between genetic markers. And this is important that's an important feature of the BLR models. And what outcomes after the BLR model analysis, what we believe is a cleaner signal. So we have identified genetic variants that are more likely to be associated compared to the rest of the variants. And that goes across the genome and across the diseases. So that's at one level. The next level is we take these adjusted summer statistics into in kind of, you can say, the biological interpretation machine. So basically what we do, we link genetic markers to information in different biological databases. It could be genes, it could be proteins, it could be medications, it could be biological pathways, and so on. And we can now interpret this adjusted BLR signal in a new way. So basically in the context of these what we call genomic features or functional marker sets. And we can start to identify patterns that are unique for some certain diseases and for certain features. We have recently published a paper on these BLR models where we show that improves prediction accuracy. In the upper left corner here, we have what is here with the methods that's named with base R, C, N, A, and L. That's basically the BLR models. And in the bottom here, we have the clumping and thresholding methods, standard procedures. And what you can see here is that the accuracies, which is shown here on the x-axis, is better for the BLR models for BMI, but also for human height. And what we believe, and we have evidence from simulations that shows that the BLR models are actually better at fine mapping. And what that means that if we are better at fine mapping the true causal variants, we can also better identify the true underlying disease biology. And if we better understand the true underlying disease biology, we could potentially also develop better drug targets. So what the current work is focused on investigating, basically validating the BLR models in terms of fine mapping and comparing to existing methods. And as I said, type 2 diabetes is correlated with a range of other traits and diseases. And here just shown a few of the cardiometabolic diseases. So coronary artery disease, BMI, waist-hip ratio, chronic kidney disease, and atrial fibrillation. And there are, of course, other diseases that are important, but what you see here to the right is the genetic correlation, and a dark red color means that it's highly correlated. So, for example, type 2 diabetes is highly correlated with CAD, BMI, and waist-hip ratio. And if they're correlated, that means we can borrow information across the traits from a genetic point of view. And where we are now is that we are evaluating both the single trait and multiple trait BLR models in terms of mapping power and precision of parameter estimates. And that's work that's in progress. But based on the prediction accuracies we've seen on BMI and height, we believe we will have increased power. Another important aspect of the project is that you actually collect a lot of different types of data. So we have set up a workflow. In fact, it's like a database you can download where we have linked genetic markers to different biological entities. So it can be genes, it can be gene ontologies, it can be protein-protein interactions, protein-chemical interaction, and so on. And a lot of in this process, there's a lot of quality control that is needed, but we have built up a framework where we can quickly integrate new types of biological information in our workflow, and all of that is available on the GitHub, and you can actually download the first version of this what we call genomic informed drug target database for type 2 diabetes. Here are just some quick results. We have done some marker set tests. So basically, we have identified different what we call genomic features that are associated with type 2 diabetes. We have genes to the left, we have pathways, gene ontologies, and chemical protein complexes. And what you can see here, for example, for the genes, there are some very well-known genes associated with type 2 diabetes. And some of these are also associated, for example, for BMI. But there's interesting patterns here, both for genes, pathways, and so on. And all of that requires us to sit down and interpret it in a biological context, and that's some of the work that's currently ongoing. We also the way we do the marker set test is a standard approach, commonly used, and we need to compare that to some of the BLR model marker set tests that we also have developed. What you can use this framework for is to test some very specific biological hypotheses. We can test one hypothesis could be that we claim that genes, proteins, metabolites, pathways functionally associated with type 2 diabetes are enriched for association signal with type 2 diabetes. That we can, with our framework and analyze both the methods we have implemented in our software, but also the database, we can actually test these directly. And there are other hypotheses we can test. Another important aspect of what we're doing in the project is we need to validate the methods. For example, how many of the previous associated genetic markers, genes, pathways, do we detect using our BLR procedures, and how many of them are new, and are they potentially irrelevant drug targets? And there's also some work that's in progress. But we have a framework where we believe we can do a lot of these things quite efficiently. And just to end up here, the project is about method development, it's about method validation, and it's also about bringing these methods out to the scientific community, both in academia and industry. So we have developed open source software and tutorials. We have released a new and updated version of the RPact QGG that include the modeling approaches that we have proposed in the BALDER project. It's again available on GitHub. We have developed tutorials so you can actually do these type of analysis at home or in your own lab. You can also, what we have also released, what I would say is a preliminary version of this genomic informed drug target database for type 2 diabetes, and there we also have tutorials for using that. All of this, I would say in particular the drug target database is a work in progress, but it's a state where I think we can share a very preliminary version of it. So with that, I have ended my talk, and I'm happy to have questions.